Hello, everyone, and welcome to GenScript's panel discussion on mutant libraries for precision engineering. My name is Dr. Kimberly Arnold, and I'm the product manager of molecular biology services at GenScript, and I will be moderating today's panel. We have with us today 5 experts in the field of protein engineering to discuss precision engineering of proteins, involvement of mutant libraries, and what the future may hold for protein engineering. But before we get into our panel discussion, I would like to hand over the microphone to Dr. Wynn, our Senior Field Application Scientist at GenScript, to set the stage for today's discussion. He will share with you how we at GenScript create precise mutant libraries for engineering all kinds of molecules, including antibodies and enzymes, and compare this to current methodology that is being used. Wynn, it's all yours.
Thank you very much, Kim. Hello, everyone, and thank you very much for being here. So for those of you who are not familiar with GenScript, we are the #1 provider of gene synthesis services in the world. We have about 3,000 employees, with many of us are PhD and master level scientists. And we also have 30,000 plus citations in peer-reviewed journals with over 110,000 customers all over the world. We make DNA and more, so when you partner with us, you level up. So GenScript essentially has 3 core platforms, molecular biology, protein, and antibody. But today I'm just going to be focusing on one of the molecular biology services. It is known as the Precision Mutant Library Service. So first, what are precision mutant libraries? Precision mutant libraries are high-quality, diverse synthetic libraries containing only desired mutants. These libraries offer even distribution of amino acids with little bias, inclusion of user-defined codons, elimination of stop codons, as well as codon optimization for specific hosts of interest for enhanced expression. So our precision mutant libraries are enabled only because of our next generation semiconductor-based DNA synthesis technology, coupled with our automation platform. Together, they enable us to precisely control incorporation of individual nucleotides into the growing DNA strand on a silicon chip. But in contrast, if you look at the libraries that are commonly constructed through traditional methods using degenerate codons such as NNN or NNK, there are a number of major issues associated with those libraries. For instance, you cannot eliminate stop or unwanted codons. There is no control over amino acid ratio. So your library is likely going to be biased with over or under representation of certain amino acids. And also codon optimization is not possible. So such lower quality libraries would require higher demands for screening. And so it's just going to cost you more downstream. With the unique qualities of our precision mutant libraries, as I presented in the previous slide, we can address and overcome all of these issues. So as we're all scientists, we would like to provide data. So this is the experiment data to support our claim. So we built 2 libraries, one using NNK degenerate approach, and then the other one using our PML approach. So in this comparison study, we also specify a design requirement, which is we only want 3 amino acid incorporation in the first mutation site, 10 in the second, and 20 in each of the 3rd and 4th positions. So as you can see here, you cannot incorporate such a design specification in the library built with NNK degenerate codons. You get all 20 amino acids in the first and second sites, even though you only wanted 3 and 10, meaning that you are, it's going to require a lot more screening efforts to cover the diversity that you want. Whereas for our precision mutant library, you can see that the amino acids are evenly distributed. And also you get exactly what you specify, 3 codons in the first site, and then 10 codons in the second site, and 20 in each of the 3rd and 4th positions. So this is actually going to translate directly into screening efficiency. And you can see here that the screening efficiencies of our precision mutant library are substantially higher than those of the NNK library. So as a result, our precision mutant libraries can significantly reduce the burden of screening and the subsequent characterization. So there are three different types of precision mutant libraries. The first one is a site saturation library. This is for those who are interested in mutating a single site to all 19 other non-wild type amino acid. So this would be saturation scanning library. And the second one is this would be site saturation library. And then the second one is saturation scanning library. This library is going to be very similar to site saturation, except that now you have more than one site to mutate out to 19 other non-wild type amino acids. And lastly, combinatorial mutant library is for those who are interested in mutating multiple sites within multiple regions, all at the same time. So simultaneous mutation. So this library is going to be substantially, the size of this library is going to be substantially higher than the other two. We build these libraries by synthesizing individual oligos carrying specific mutations and assemble them into double-stranded DNA format or clone them into a plasmid of your choice. So we can deliver two different formats, either double-stranded DNA or plasmid format. And I would like to stress that, unlike our competitors, the diversity of the library is not going to be compromised due to cloning, for our capabilities lie not only in the DNA synthesis, but also in assembly and cloning. Also, our error rate is extremely low. It's 1 in 3,000 base pairs. And our minimum coverage guarantee is 90%. And we also do NGS as part of our QC, and you will get a report upon delivery so that you can also look at the amino acid distribution in your library. So I'm going to give you three case studies today. So this is the first case study. This is for enzyme engineering. So the customer has 484 amino acids that they would like to improve the enzyme activity. So they want to target three different regions. The first region has 14 sites. The second one has 12, and third one has 12 as well. So this is a saturation scanning mutagenesis library, meaning that all the mutation sites are going to be mutated out to 19 other non-wild type amino acids, generating about 722 mutants, and the customer want all the mutants to be delivered in a single pool. So like I said, we do NGS as part of our QC. And as you can see here, this is the NGS data. And for every single mutation site within the three different regions, we were able to achieve the replacement of all 19 other non-wild type amino acids. And the yellow blocks are actually wild type amino acids. So we were able to achieve 100% coverage in this case study. This is a more visual representation of the previous data. And as you can see here, each color block is an amino acid. So for all mutation sites, we will able to achieve very evenly distributed amino acids. So this is a library of superior diversity, where the library is not going to be underrepresented or overrepresented with certain amino acids. So unbiased libraries at the end of the day are going to help you minimize screening burden, because every single mutant in the library has equal opportunity to be selected as a top candidate. And this is a second case study where the customer want to improve the function of the capsid protein. So there are 4 positions that the customer want to target specifically. And they have 7 specific amino acids that they would like to use as well. But they want to mutate these all 4 of these positions simultaneously. So this is going to be a combinatorial mutagenesis library. And this will generate about 2,400 mutants. And the customer also requested codon optimization for E. coli and to have the library delivered as a single pool. So the outcome was we were able to successfully construct the desired combinatorial mutagenesis library with 100% coverage, incorporating all customers' requests. Last but not least, this is a case study for antibody engineering. So The customer would like to improve the affinity of the antibody that of their interest, and they want to target 6 different CDR regions, as shown here, with a total of 63 sites. Again, this is also saturation scanning mutagenesis library. The customer wanted to mutate all 63 sites to 19 other non-wild type amino acids, but one site at a time. This generated about 1,200 mutants, again, to be delivered in a single pool. So this is the NGS data again, visual representation. And as you can see, the amino acid distribution is very even for all mutation sites. So in this case, the customer actually shared the data with us. So the customer used their proprietary screening and characterization method. And in this chart, any value above 1 is considered better than wild type. So you can see that there are quite several top candidates, resulting from our library. The customer then chose a subpopulation of these candidates for further characterization for their binding affinities using surface plasmon resonance binding assays. So as you can see here in the chart, in the table, there are 5 beneficial mutations. These are the mutant sequences with improved affinities, affinities that are substantially higher than that of the wild type, with one particular mutant in the femtomolar range, 4 orders of magnitude higher. And as a second step, the customer wanted to explore the combinatorial effect of these beneficial mutations. So we helped them generate a combinatorial mutagenesis library combining all of these beneficial mutations. And then the customer screened them again. And from the second library, they were able to identify 3 combination sequences that show further improved affinities compared to the affinities of their individual parent sequences. So I shared 3 different case studies with you guys in this presentation, enzyme engineering, antibody engineering, and viral vector engineering, but our precision mutant libraries can be applied for any type of protein engineering, as well as even for promoter engineering. And you can imagine, this type of application is going to be very useful for someone who is building a genetic circuit or doing a pathway engineering. Because the expression of different genes within the circuit or the pathway has to be coordinated to generate the best possible outcome. So in summary, our precision libraries can be utilized for a variety of engineering disciplines for various applications. And so this actually is the end of my presentation, and I thank you very much for your time and attention. Now I yield the floor to our panel discussion on mutant libraries for precision engineering. And I'm very excited to have our 5 leading protein engineering experts for the panel discussion today. Kim, please take it away.
Thank you, Wynn, for that great presentation. We're now going to move on to our panel discussion. We'll also take questions from the audience following our discussion, so please feel free to type your questions in the Q&A box. Let me first introduce today's panelists. We have Dr. Grant Murphy joining us today. He is the Director of Protein Engineering at Merck & Company, where he leads a team of scientists working at the interface of chemistry, biology, and engineering. The Merck Protein Engineering Group has engineered dozens of enzymes and therapeutic proteins to enable drug discovery, development, and clinical supply, and is actively engaged in developing new protein engineering and computational methods. Our next panelist is Dr. Vesna Mitchell. She is the Senior Director of Molecular and Cellular Engineering at Codexis. She leads a team of talented and enthusiastic scientists responsible for implementation of code evolver technology across Codexis's protein engineering programs, with a focus on high throughput library generation, next generation sequencing, library design, and optimization of protein expression in multiple host organisms. We also have Dr. Nikki Kayaza today with us. He is the CSO and co-founder of Celebri. He's a biotechnologist with more than 10 years of industry experience, bringing with him significant expertise in microbial genetics, cellular physiology, and synthetic biology, with applied technical expertise in metabolic engineering and biocatalyst development. Our next panelist is Dr. Po-Soo Wong, who is an assistant professor at Stanford University. His research focuses on advancing the understanding of proteins for the engineering of novel therapeutics and other protein-based nanotechnology. His group uses computational modeling, structural biology, and experimental library optimization to create novel molecular platforms and advancing AI tools for protein design. And finally, we have Dr. Justin Farlow. He is the co-founder and CTO of Serotinib, a synthetic biology company using software to engineer at scale new cell therapies against cancer, focusing on engineering proteins that are deployed as functional payloads into cell-based therapeutics. His company couples high throughput assays in the wet lab with AI-guided protein design to rapidly explore a massive landscape of novel cellular functions. So I want to thank you all for joining today's panel, and please turn on your video cameras at this time. And when you can stop sharing your screen. All right. So we're going to get started today with some questions. The first question I have is for all of you. Wynn just presented how to apply precision mutant libraries for different types of engineering. And given everyone's diverse background, can you please share your insights around how these libraries can be applied in other segments of synthetic biology, perhaps in regard to your own research efforts? Nikki, can you get us started?
Yeah, absolutely. You know, as a metabolic engineer, we're often looking at, you know, proteins as the integral role or integral part of a pathway to produce the products that we want to make using our cellular factories. And so we've had to improve a wide range of properties of proteins or enzymes in particular for our pathways, whether it be finding an enzyme that you like that has the activity you want, but you need to improve the temperature tolerance of that enzyme or improve the catalytic turnover of that enzyme using protein engineering and library designs, or the pH tolerance of that enzyme. Not only that, but we often look at the product selectivity and specificity of the enzymes, which enables us to potentially link novel pathways together that may not exist in nature. There may not be an enzyme that can bridge two pathways, and by finding an enzyme that can utilize a similar product or similar substrate or produce a similar product, we can use protein engineering to then create novel activities that can then bridge those two metabolic pathways so our cellular factories can take simple feedstocks and produce novel compounds.
Thank you, Nikki. Grant?
So we use precision libraries in a way very similar to what Nikki described. I guess one area that I would highlight is we often use computational protein design to whittle down the set of mutations we'd like to make, and then use precision library techniques to incorporate those, moving away from degenerative codons. And I think the hope there is that our computational techniques will get sophisticated enough and combined with AI that in the future we're making orders of magnitude fewer mutants, and hopefully in the future just one.
Okay, sure. Thanks, Grant. And Po-Soo, do you want to comment, please?
Yeah, it's very similar to what Grant was saying. So from a different perspective, since we are mainly developing tools for these protein design and not necessarily just with the goal of engineering useful mutants, one thing we noticed that with this type of precision mutant library, what we could do is that if we form a hypothesis and we build computational models of these proteins, the fact that you can cover exactly the distribution of positions and mutations that you design into these proteins, you could actually feedback on our hypothesis much better than doing dealing with like a degenerate codon, because sometimes you get a beneficial effect and then we would have a hard time explaining it if it actually wasn't part of the original hypothesis. But of course the most useful point here is also just that oftentimes these computational designs make a very large number of diversity, and it will already push the limits of our experimental capability, and having the ability to reduce the library size is extremely useful.
Great, thank you. And Justin, can you provide some insight?
Yeah, so what we do is often a little different here. And so we actually have coupled this precision mutation along with combinatorial design of these proteins. And so we're trying to build proteins that are multi-domain proteins that are often large, transmembrane, unstructured, and for purposes of altering a cell's properties rather than looking for a KD or KCAT. And these large libraries, though they're large, as we mentioned, for experimental purposes, from a computational perspective, we've winnowed down to, in our case, hundreds of thousands of variants of different arrangements of protein modules, of protein domains. And these large libraries allow us to explore that space in a targeted and systematic way, which again, as Posu mentions, allows us to then bring software onto the analysis side and figure out which of those changes, perturbations we're making to the protein have what effects.
Great, thanks. And then Vesna, can you provide your perspective?
Sure. So at Codexis, we engineer and develop novel proteins for biotherapeutics, pharmaceutical, life sciences, chemicals, food and feed, and many other markets. Our technology relies on iterative cycles of diversity generation, high throughput screening and sequencing, and then use of machine learning and recombination of beneficial mutations to improve multiple properties frequently on any given program. Mickey gave examples of improvement in specific activity, pH tolerance, solvent tolerance, cofactor utilization, on some of our therapeutics programs, reducing immunogenicity. So you can imagine that we really like to know what's in our libraries. We also like to reduce our screening efforts and get the maximum amount of information. So Posu also brought up the fact that if you have random changes in your libraries, you can't quite learn as much from them. So precision type engineering is important for us. It also helps when we're screening in difficult cell lines, for example, or with expensive substrates. So if we can screen less and learn more, that's always a good thing.
Yeah, definitely. And you actually segue into my next question. You just brought up, I'll address this to you. What are the key advantages of using these precision engineering methods, like these precision mutant libraries, over a randomized approach in biomolecular engineering?
So I already said it, if you have a high quality library and you know exactly what's in it and there's very little bias, you can reduce your screening efforts. That screening effort is costly. Not only is it a cost of your substrate and your consumables, but also the cost of your time and instrument usage. So if you can somehow increase that amount of information that comes out of your screen and free up time to do other screens so you can learn more, that's always good. So if you don't know anything a priori about the effect of mutations, then typically we like to look at every possible mutation. If we know that there's only 20 possible changes in that library and we don't have to screen much more than that to obtain the desired coverage, that's good. Also, random mutations are rarely serendipitously beneficial. Most of random mutations will kill your library. So if you can avoid those, that's also good. Last but not least, some of the traditional random approaches that I personally haven't done in many years are very limited in the amount of diversity you can access. So in error-prone PCR, that is still being used in some protein engineering programs that we read literature about, for example, you can only access 6 or 7 changes from any given codon. And if you target one change per KB, then 30% of your library is the wild type sequence. So you have to screen many, many redundant parental sequences to find only a handful of changes that may or may not be beneficial. So precision engineering actually highly improves the library quality.
Okay, yeah, you hit a lot of the key advantages. Does anybody have anything else that they would like to add to Vesna's comments?
I think I would add that using random libraries, it's very hard to interrogate a hypothesis. And if you have multiple different hypotheses, so using a precision library, you can target a library for each of those different hypotheses and then extract out maybe what features of your system need to be improved.
Okay. Yeah, definitely. Thank you, Grant. Anybody else? Nikki?
Yeah, I mean, I guess I would only, I mean, I agree with everything that was said. You know, as a small startup, you know, resourcing is key. And, you know, when we look at protein engineering and metabolic engineering in particular, trying to alleviate a bottleneck through protein engineering often creates a bottleneck in the lab in terms of screening and fermentation. And the ability to now take, you know, the number of clones that you would have to screen in a random library and now be able to look at 2 or 3 different proteins based on library size, because we often have multiple bottlenecks in a pathway. Once you solve one bottleneck, you have another bottleneck that immediately pops up. And so it's a constant cycle of trying to improve an enzyme. And again, the less you have to screen to do that, the more ground you can cover in the same amount of time.
Yeah, I agree with you. Posu, Justin, any additional comments? No? Okay. And so, you know, a question here, you know, about COVID. We have a lot of synthetic biology efforts being placed in the development of therapeutics and diagnostics for COVID-19. Could you provide your insights on how precision engineering using mutant libraries may be used in such development? I'm going to direct this question first to Posu. Do you have any insight onto this?
So my lab don't specifically work on the COVID part of the target, but one thing that we do use, and that was related to the discussion earlier and also perhaps could actually be extended to the COVID problem, is that when we have an experimental system where the protein engineering is actually tied into a set of barcodes. So it's not just that we need mutations on the proteins themselves. They do have to match up with a coverage barcode where we can read out the identity of those proteins later. And it turns out that when we were trying it, this precision mutant library, just because, you know, from our experience, actually, the quality actually is incredibly high. So our coverage of matching mutations, you know, you have a set of mutations somewhere, and then matching random barcodes. The matching coverage when we get from the NGS was exceptionally high. So we were able to actually see every count of our mutation and also every combination of the mutation that we put into the original protein, and that is incredibly beneficial. So you know, how does this tie into COVID? Basically, when we try to use this assay, and then the assay is to screen for protein-protein interactions. So essentially, if you do a library-on-library selection of protein interface, right, you would have a set of protein on one end, the other set of protein on the other, and both are barcoded. And the fact that you have this precision coverage with the barcode matching to their mutations, you can screen for interface. And then oftentimes this is a critical step for, you know, finding engineering protein interfaces and designing inhibitors for viruses. So that's our experience with this type of approach.
Okay, yeah, thank you. Grant, do you have any insight into this? Oh, you're muted.
Thank you. For COVID especially, time is of the essence. And so that low error rate means when you pick 2 or 8 clones to verify the sequence, you have a really high chance of getting the clone you want. And every time you have to go back and repeat that process, that's another day that a patient is waiting. So, you know, I think for COVID, it's really around time. I think for all programs, that's true. You know, every program is the most important program, so they say. But the low error rate also means that you can take what previously was a mutagenesis experiment, and you can think about it as a de novo gene assembly experiment, or you can start to think about how do I incorporate multiple mutations simultaneously. Vesna mentioned that in random error-prone libraries, it's very hard to generate sequences with many mutations simultaneously. In this case, if you have lots of oligos with very, very low error rates, you can incorporate lots and lots of mutations simultaneously that you know should be beneficial. And then that reduces your screening burden by giving you a larger pool of beneficial sequences.
Great, thanks. And does anybody else want to add to any of the comments from Grant or Posu? Okay, yeah, thank you guys for your insights, especially with this pandemic. It's a time is of the essence to get these diagnostic and therapeutic tools out. Moving into AI, we see a lot of libraries users inquiring about our precision mutant libraries for AI purposes. Do you think AI will be the next big thing for protein engineering? And I'll turn this question over to Justin.
I think AI is going to be very powerful in protein engineering. AI needs data, and what these libraries allow us to get is a lot of data, and it allows us to get data that is targeted. In our case, our software we built in-house allows us to learn from each of these designs, and so the more designs we have, and the variety of those designs, especially if they're, as was mentioned, coming from a hypothesis, the better our machine learning algorithms get. Part of what makes AI or machine learning useful to us is that it allows us to find subtle trends that are tricky for a human to pick up on, especially if there are many of them. And in our case, we're looking at the outcome of a mammalian cell based on the subtle changes of a protein. And there's a lot of distance between there. And so having a large library of variations in arrangement and of presence of particular protein domains allows us to get downstream to that final question, again, using machine learning, of whether or not we're increasing the efficacy or safety of a cell therapy. So these large libraries that GenScript help us build allow us to get the kind of data and the quality data that we need to ingest into these machine learning applications.
Great. Posu?
Yeah, so with AI, there's, I would say there are at least two different flavors of this. So generally what people do these days in terms of protein engineering, it could just be a very limited case, so other people can correct me, is that if you focusing on a family of proteins, and then if you can do a pre-screen on a small set of libraries to get some information, the machine learning code can extract the information really well, and then you can move on from that and reduce your number of steps for selecting for properties. But my lab does a slightly different flavor. So we don't operate on the sequence level, we operate on the protein structure level. And with that application, essentially what we are doing is something like molding a protein structure against a target, like say, for example, constructing antibodies where the loops can be directly targeted towards an epitope of choice. And through this practice, what we realize is that the computer code, so what we were trying to handle is the flexibility and the dynamics of the structure. But, again, I had mentioned earlier that, through these machine learning models, accounting for this flexibility, we have a ton of solutions. And we're extracting consensus through the AI algorithm, but then in order to actually test them, narrowing it down to very specific epitope where our antibody design efforts are hitting, actually really needs something like this precision library, because when we design, again, this is the testing hypothesis, like, were you able to make a certain contact with a certain set of amino acids? Were you able to make a new, shape complementarity with a different pocket of a protein? There are multiple sites on the protein that we have hit at the same time. So if you don't have an ability to actually reduce the complexity for each one of those sites, usually the target just is kind of exploding in terms of the combinatorial complexity. So with our AI approach, just because we're actually expanding looking into details instead of synthesizing consensus. But even in those cases, it's extremely useful for us.
Okay, yeah, thank you. And we have time for one more question for this panel discussion. What do you envision as the future direction of synthetic biology in regards to protein engineering? And I'll have Grant start us off.
So I'm gonna lay out a bold answer here. I think we're moving towards a post-evolutionary world where the sequences, the cofactors, the functions that we're using in protein engineering will be not derived from nature. They will be created entirely from scratch using the types of work that POSU does, and that will find its way into synthetic biology, and we will engineer outside the confines of biology. In vitro transcription translation systems, novel parts. That's the future.
Okay, and POSU, do you want to continue?
Sure. I mean, this is definitely debatable. Like, everybody has a different opinion on like where the future of this protein engineering is. It seems like what's really exciting here is that the lines becoming blurry, right? Like, it used to be there's a rational design and then there's like direct evolution, but then through machine learning efforts, the rational design is I don't know if you would consider that rational design, but the way to come up with synthetic sequences is already blending in with this direct evolution approach. And then now I think with new effort like what we're trying to create, to even merge further with the structural information. You know, it's like the protein design, direct evolution, and I guess, well, there's like the component where you have this random direct evolution through rational design combining with structure is all merging in recent days. And I think that's a very exciting time to go forward. So.
And Justin, do you want to continue as well? Add your
I'd say I see all those things happening, and they're very exciting. I think the one thing that we do at Seratni is to try to take all of those components as domains, and rather than designing proteins based on mutation space, but actually based on domain space and the mixing and matching and arrangement, much as biology has done with recombination, and exploring how the combination of both wild type domains as well as these novel domains, when you put them together, you can build something yet different. And I think part of that is the ecosystem to understand the way those different components, either in one protein or between proteins, interact has been tricky to come by. But now we have the bioinformatic tools, again, the cheap and economical DNA synthesis, as well as kind of the high throughput characterization loops that allow us to build on each of these discoveries to push protein engineering farther.
Great, and Nikki?
Sure. You know, I think there's, you know, the aspect of even just synthetic biology tools, reagents, applications, instrumentation, you know, enabling us to get better and better at screening these libraries that are being produced, and closing the circle between testing the hypotheses with positive data, negative data, to build better statistical models, homology models, you know, et cetera, to inform that. And I think, as Grant mentioned, sort of going beyond the confines of life. But in addition to that, you know, if you look in any genome, still 40–50% of the proteins there are unknown, right? What do those proteins do? Well, you know, AI protein engineering, these models help us, you know, better understand, you know, the unknown proteins and enzymes inside cells that will provide potentially novel cures, novel targets, novel medicines, you know, et cetera, as we better understand, you know, life as it is in an organism or in a cell.
Yeah, definitely. And Vesna, final comments.
I won't repeat any of the comments that, you know, I very much agree with. I would like to add that, you know, these advances in synthetic biology push protein engineering, and as we learn more and more about each protein, we learn about interactions between different mutations, interactions between different domains. We can also push the progress a lot faster, right? So I'm really excited. I've seen, you know, the shrinking of the timelines for protein engineering projects that we have done even in the last couple of years. because we have more and more tools from synthetic biology, there's also been a lot of development in machine learning and AI, bioinformatics, other bioinformatics tools that really enable us to make more and test more hypotheses in a much shorter time. So I think that's probably the most exciting part for me.
Okay, great. Yeah, thank you all. This ends our panel discussion for today, and now we're going to open up the remaining time for some audience questions to our panelists.